Hello, my name is Chad Kim. I'm a postdoc in the Spiegelman lab at Dana Farber Cancer Institute and Harvard Medical School. Today, I'd like to talk about irisin, endurance exercise-induced myokine in skeletal muscle. I'm going to talk about its roles in bone and its mechanism. Exercise affects many organs with beneficial effects. Exercise improves bone strength, lowers fat mass, and enhances the functions of liver and muscle. However, these effects vary based on the type of exercise. In bone, resistant exercise improves bone mineral density. However, endurance exercise causes complex outcomes. Swimming and cycling don't raise the bone mineral density, while running improves it. A recent paper showed that endurance exercise causes bone resorption to support calcium ions. That means we need to consider that the outcome of endurance exercise is very complicated. This different outcome between resistance and endurance exercise can be explained by the components of exercise. Resistance exercise uses mechanical stimulus relatively more, while endurance exercise uses less than mechanical stimulus, but probably they may use more biochemical communication, and that cause maybe different genes and protein level changes. Among these biochemical stimulus, exercise-induced muscle hormone or myokines have been studied to understand this phenomena as the tissue crosstalk between muscle and bone. Irisin is one of the myokines for this tissue crosstalk. Its precursor form is FNDC5, which is type 1 membrane protein staying on the plasma membrane in skeletal muscle. Endurance exercise increases the expression. It is cleaved to relieve a soluble form, which our lab named irisin as the messenger protein. This protein gets into blood to reach other organs. According to absolute quantification using mass spectrometry, its concentration is 3 to 5 nanogram per milliliter in human plasma, and endurance exercise increased the levels. Increased irisin in blood reaches to other organs. Initial studies show that irisin increases thermogenic program in fat, which can increase energy expenditure. In bone, low dose and low frequency of irisin injection increases bone formation according to Dr. Grano's group in Italy. But their injection with higher dose increases bone resorption, which we will explain here. It is also known that irisin improves cognition. To understand the functional roles of irisin in bone, we used FNDC5 knockout mouse model, which lacks irisin as FNDC5 is the precursor form. Here we performed ovaryectomy, which causes bone resorption because of lack of estrogen. This surgery mimics a post-menopausal woman as the population has high incidence of osteoporosis because of that lack of estrogen. Left are representative figures of histomorphometry of backbones from sham or ovaryectomized wild type or FNDC5 or irisin knockout mice. Histomorphometry stained bone mineral as black, so you can see black stain line which are bone mineral area, and inside of that outer layer of the bone we call trabecular bone here, and we analyze that trabecular bone area. As you know, wild-type mice after ovaryectomy has less black stain in the trabecular bone, meaning that less bone mineral compared to sham mice. However, FNDC5 or irisin knockout mice doesn't show any difference between the sham or ovary ectomized group. Right are the graphs of analyzed data from histomorphometry. As you can see, these knockout mice have more bone volume and trabecular bone compared to wild-type mice in ovary ectomized group. The lower panel, the graphs also show that ovarectomy increased eroded surface and osteoclast number in wild type. Here, osteoclast is performed resorption cell. But in knockout model, there is any increased level of eroded surface or osteoclast number. These data indicate that FNDC5 or Irisin knockout blocks ovarectomy-induced bone resorption. So next question is, which bone cells respond to irisin? Osteoblast works for bone formation, while osteoclast cell work for bone resorption. And these cells were regulated by osteocyte. Osteocyte is the main bone cell in bone, about 9%. And the osteocyte responds to biochemical or mechanical stimulus. And upon that signal, osteocytes secrete the proteins to regulate the function of osteoclast or osteoblast cells. So we tested first whether that irisin can help the cell survival, that osteocyte cell survival, because that open that abnormal that osteocyte cell death is the cause of osteoporosis. So that's the reason that the cell survival is very important for bone homeostasis, and we want to test whether irisin can contribute to that bone homeostasis through that. And second thing is that we also like to see whether that irisin regulates osteocyte function, here secreting factor. With the collaboration with the Linda Bonewald group, we used the osteocyte-like cell line, here is called MLOY4. And when we treated hydrogen peroxide, which drives the cell death by oxidative stress. But when we treated irisin with that hydrogen peroxide, as you can see, that cell death rate decrease. And that decrease begins with 1 to 10 nanogram per milliliter of the irisin. As I mentioned, that this is the range of the physiological concentration of the irisin in human plasma. So that means probably irisin may improve osteocyte cell survival in vivo. We also tested secreting factor, here sclerostin. This gene is mainly expressed in osteocyte, and the level increase upon exercise to increase bone resorption. As you can see, irisin treatment increased the expression of the sclerostin in osteocyte-like cell line, as you can see left. The middle is the mouse model study. I did irisin injection, daily injection for 5 days, and I collected the osteocyte-enriched bone from tibia. And I look at the gene expression of sclerostin, showing that irisin increased their gene expression like this. The right panel is the measurement of the sclerostin in blood, because that secreting factor eventually get into the blood, so you can measure their protein levels, and that outcome match to their gene expression study. We further determine its mechanism, especially we want to discover the irisin receptor. So we use the osteocyte-like cell line since that cells respond to irisin. So I treated cell with irisin or adopsin, another circulating protein, as a negative control. And then we precipitate this irisin or this adopsin after cross-linking on cell surface, and we look at which proteins enriched with irisin compared to that adopsin. And this is table showing, this table shows top 5 cell membrane protein we found. And among them, we're trying to narrow down the receptor upon as the cell membrane protein or any signaling protein upon ligand binding. And that category just comes to only integrin beta 1 here. So what's the integrin? Integrin often form heterodimer with alpha and beta together. And there's many integrins known, and 24 pairs of the integrin have been reported. And one of the features of integrin is that the ligand bind to multiple integrin pairs. So we'd like to know that whether irisin binds to several integrins. So I did binding assay with the irisin and solver integrin. Here's the active domain integrins. And we look at their binding. As you can see that integrin alpha 1 and beta 1 bind to irisin. However, here alpha V and beta 5 has more affinity toward the irisin. That means maybe you can use that active domain of integrin alpha V beta 5 pairs for any biochemical and biophysical study. So we did some biophysical study to determine their binding motif using unbiased methods here, hydrogen deuterium exchange and mass spectrometry. That data show that the irisin and integrin actually bind, and we determine their binding motif. Especially on the side of integrin beta 5, only beta A domain is determined as the binding domain toward irisin among whole active domain. Also, we can determine their binding motif, which is already known for ligand binding side. So that means maybe irisin bind to integrin as the well-known traditional integrin ligand binding pathway. So we look at what's the outcome upon their binding, and their traditional ligand binding toward integrin often causes the signaling, and the signaling begins with the phosphorylation of FAK, and their downstream begins after that. So we look at the level of the phosphorylation of FAK and their downstream. As you can see that irisin treatment on this osteocyte cell line increased that phosphorylation of FAK. Also, you can see other downstream also increase their phosphorylation level upon irisin treatment. Furthermore, I'd like to know whether that integrin mediate irisin effect in the cell. Here is the sclerostin gene expression. As you can see that the irisin increased sclerostin gene expression in this cell line, but when I treated that cell with the well-known iris integrin and ligand binding inhibitors, the irisin mediated or induced gene expression are blocked. Also in vivo model, wild-type mouse can get irisin with this one of the inhibitor, showing that integrin ligand binding inhibitor block irisin induced sclerostin gene expression as well as the protein level here. So as a summary, we can say the osteocyte responds to the physiological concentration of the irisin. And integrin alpha V family mediates that the irisin effect in osteocyte, and irisin prevents that hydrogen peroxide-driven cell death of osteocyte, and irisin increase the sclerostin gene expression in osteocyte, and that sclerostin is known to cause bone resorption, which matches our observation in vivo in knockout model. As a future plan, we would like to know what's the bone phenotype with longer term exposure of irisin. We are working with Cliff Rogen and Laurent Bernard for this study. Also, we'd like to know whether we can use this knowledge to find a therapeutic approach toward osteoporosis, because that knockout of the irisin completely block overectomy induced bone loss. So if we block irisin chemically, we may block some osteoporosis here. So we're trying to use making the antagonistic monoclonal irisin antibody, then we can use that antibody to block irisin function in bone resorption. Also, more mechanistic study, we try to understand that integrin, how integrin working with irisin by finding their cofactors. Also, I'm working with Joshua Cho lab to understand what's the role of the irisin toward osteocyte in space or microgravity, and we like to understand that whether irisin is involved any bone loss in the space trip. And because irisin is working for bone resorption, so we try to get any blood sample from astronaut to measure irisin level, and they can give us some kind of idea that whether irisin is actually involved in that bone resorption. I'd like to thank my mentor, Bruce Bigelman, for his support and his mentoring. And also I appreciate our lab members for many help. I especially thank our bone collaborator, Cliff Rogen, as the very good collaborator, but also he taught us how to do that bone study. So it was very grateful time being with him, and we like to work with him more. And then Lauren Barros helped us to analyze our bone, and also Mary Buxian as well. Also, to understand osteocyte, we need tons of help and also tons of teaching, which is done by Linda Bonwart. We thank you for your attention, and I'm happy to take any question.